Next up is Will Hazard with 73 Medical.
Thank you, Erica, and on behalf of 73 Medical, I'd like to thank the Penn State community for the opportunity to talk to you. About a less invasive, cost-effective solution for the treatment of hydrocephalus. Well, why hydrocephalus? Hydrocephalus is a disease that affects all ages. In the pediatric population, approximately 1 in 500 children are born with hydrocephalus. In the elderly population, it's often misdiagnosed as dementia, and there's upwards of 500,000 patients in the U.S. alone that have this condition. The treatment for hydrocephalus remains to this day exclusively a surgical implant. Basically, it's a 6-inch tube that goes through your brain. It collects the extra fluid, and with a longer tube, it puts that fluid into your belly. The problem with this device is that it has the highest failure rate of any surgical procedure performed in modern medicine, the highest failure rate. There are currently 1 million people throughout the world with these devices in their bodies. VP shunts, not hydrocephalus, but VP shunts cost the U.S. about $3 billion a year. This is likely very low estimation, and as you can see from the graphic that this number has risen and will continue to rise as the number of these shunts that exist throughout the world. At 73 Medical, we have a solution to this. It's small, it's easy to implant, it's easy to service, and it works by recreating the normal flow or cerebral spinal fluid in the body. Well, let's talk about the key value proposition of our device. The device costs nowadays, those tubes, valves, and more tubes, cost about $120 million. The problem is they end up costing the U.S. market about $3 billion. And the problem with that is they just don't work. You have a third of them failing the first year, half of them failing by the second year, and almost all, 98% of them, fail by year 10. So they're constantly being surveyed, and they're constantly being replaced, tying these patients to the healthcare system. Our value proposition is so great that we could double the COGS of the device itself and still end up saving the U.S. market over $1 billion a year. So where are we today? We have completed large animal trials. We've proven efficacy of the device. We continue to prototype and iterate. We have patent protection. Our next steps are to continue to revise and iterate the device itself, also creating the tools necessary for the safe implantation. FDA strategy depends a lot on a pre-sub meeting coming up in early 2020. We think it's a de novo 510K product, but we want to be sure. And we're going to do it with an experienced leadership team. 2 of the companies that we've formed in the previous 5 years have raised almost $6.5 million of private equity. Previous company, which just sold last week, won this VIP tournament 2016, just sold for multiples of that. Thank you all for your time, and I'd be happy to take any questions you might have.
What type of regulatory pathway are you looking at?
So we believe this will be a de novo 510K product. All the previous VP shunts are 510K products. Ours is different. It's a different pathway. It's a different location treating the same disease. We believe the FDA will label this a de novo 510K.
So my follow-up is how much and how long?
There you go. That depends on what the FDA has to say. If it's a 510K, I think 3 to 5 years and about 3 to 5 million in raise. If it's a de novo 510K and not a PMA, I think you're not going to double those figures, but you're going to get pretty close to that.
So you explained that this is treating a disease where the current solution has an extraordinarily high failure rate. Less clear to me was, so how have you avoided those failures?
Yeah, that's great. We're not circumnavigating the way the body treats excess fluid. So current treatments take the fluid out of the collections of CSF and move it. We're taking the fluid out of a different space called the subarachnoid space, which contains about a third of that fluid. This is the way the body does it. It takes it out of the subarachnoid space and it puts it to large veins in the brain. And that's what we're recreating. So we don't have any of the major failure points to the VP shunt, which is this collection of cells called the choroid plexus that get into the shunted tubing itself and plug up not only the tubing, but the valve itself.
In your current market size assumptions, is that including only the pediatric population?
That's the US shunt population.
US shunt.
And that's likely a dramatic underestimation of the population.
Do you have, I'm sorry, go
No,
ahead.
go ahead.
Do you have any animal data on efficacy?
Yes, we used a rudimentary prototype, and we did a series of ovine. There's a hydrocephalus model in sheep that's been actually well studied. We've had those trials at the large animal facility at Penn State. So we concluded those. The first set of trials, I'm not saying that we've concluded all animal trials, but
And what type of success rate did you have? Because your claim is the current standard has a very high failure rate, I think in the 90s, you said.
98% at 10 years, a third fail within the first
So
year.
with the sheep, how did you do?
Well, I mean, I don't think you can extrapolate that data on long-term survival. Our survival was 14 days, so
Oh.
I don't, unless the catheter was malpositioned or the valve itself fails, typically these devices fail a little bit later, the BP shunts fail a little bit later. So long-term efficacy trials we have not done.
What type of changes did you have to make between your initial prototype and now you're working on your final prototype?
No,
Is that-
I wouldn't call it final by any
Not
means.
yet, okay.
A lot of the work that we're doing is around the tooling. One of the things we want to do is to make this procedure be able to be performed by people with less skill. Obviously, in sub-Saharan Africa, you can't have a pediatric neurosurgeon performing surveillance on these. So a lot of the efforts that are going into are safely placing this into the location, both in the sinus side and also the subarachnoid side, the two spaces where it invades the coating.
Are your plans then, when you meet with the FDA to determine what your next steps will be? I guess you're assuming that you're going to have to obviously do clinical trials.
Yes, yeah.
In your current
and also
assumptions.
the FDA will have a large part to do with how much money we raise. We certainly don't want to underfund the initial Series A.
And you'll plan to have the final prototype for the clinical trials?
Yes, I mean, the
You won't be
design,
changing.
yeah, the functional design will not change.
Okay.
The location of where it goes in and where it feeds into will not change. We really want to make it simple and easy and have a very high safety.
So are you looking at becoming a device company?
Yes.
Or to license the technology?
We have experience in medical devices. We would like to bring this product to market.
In terms of your team, then, you have all the expertise that you need, you think, to be able to do that at this
For
point?
this stage, we do. We obviously need help with program management and also business and finance, for
Perfect.
sure.
Okay, any other questions? Thank you, Dr. Hazard.
Thank you.
Thank you.